Alright, in this section of our biomolecule or macromolecule video notes, we're going to be focusing on lipids. Last time we got an intro to what different naming parts means of these biomolecules. We started looking at dehydration synthesis, and then we started really focusing on carbohydrates. So in this one we're going to continue with that information. We're going to look at how dehydration synthesis works in lipids, and we're going to look at types of lipids, their parts, and those types of things. So let's go ahead and get started with lipids. Alright, as far as characteristics of lipids goes, we know lipids are nonpolar. Remember when we talked about water in the last unit? Water is a polar molecule. It has a slightly positive end, a slightly negative end, kind of like a magnet almost. That gives water its unique characteristics to be a universal solvent. Most particles, most substances fall into that category of some range or another, so it gives water its ability to dissolve a lot of different things. Lipids are going to be considered nonpolar, so they don't have that slight polarity to them on either end of a molecule, and so that makes it really difficult for water to dissolve lipids. So lipids are going to be insoluble because of that. Remember our carbohydrates. Carbohydrates are a short-term energy. We get starches and sugars, and it gives us quick energy. Our lipids are going to be long-term energy. Our lipids are going to help us store long-term energy for long-term use, kind of make a reserve almost of energy when we need it, and for long energy productive processes, whether you're working out or whatever it is that you're doing, and you need that long energy storage tapped into. That's what these lipids are going to be for. Some common names for our lipids would be fats or waxes. So most times when we're referencing fats, we're talking about lipids. Alright, the building blocks of the lipids are going to be glycerol and fatty acids. So a glycerol unit over here on the left and a fatty acid unit. This is a yellow horizontal chain. These are going to be our two building blocks of our lipids. Okay, so let's get into categories of lipids. We have saturated and we have unsaturated. Saturated lipids or saturated fats are fats that are solid in structure at room temperature. Okay, think of all the things that maybe you eat or that is cooked with. Think about all of the fats that you use, and maybe you can kind of identify some that are saturated fats. Say you like steak. You get a steak at a restaurant. There's going to be fat on that steak. That fat on that steak is solid. That's a type of saturated fat. Butter that you put on toast or that you put on biscuits is going to be saturated fats. Most of your animal fats are going to be saturated. These are considered the bad fats. It's harder for our bodies to process them. They carry a lot of unwanted things. They cause your cholesterol to elevate. They could cause artery blockage, those types of things. So they're going to be considered the bad fats. The other category of these unsaturated fats are going to be more considered good fats. You need fats in your diet. You don't want to go excess in fats in your diet. And you want to make sure that you're taking in those good fats. So these unsaturated fats that are liquid at room temperature would be the good fats. Cooking oils like olive oils or vegetable oils would be an example of that. Fish oil will be an example of that. Alright, so let's look at types of lipids. We looked at the categories, we looked at functionality, characteristics, so let's look at types. The first type we have right here are called triglycerides. Triglycerides, this is a very common type of lipid, very common as far as identifying the structure of the lipid. A triglyceride contains a glycerol, so the glyceride name, it contains a glycerol. Tri means three, so it's going to be a glycerol and three fatty acids. So if you look at this picture down here at the bottom, here's our glycerol, this vertical gray chain, this 3-carbon chain. Notice the carbon, so 1, 2, 3, so a 3-carbon chain. This is our glycerol. And then it contains 3 fatty acids. Okay, so that yellow, really long carbon chain, look at all of the carbons that build up this chain of a compound. This is your fatty acid. So you have 1 glycerol, 3 fatty acids. Really easy to identify this when you're looking at pictures. Notice that this triglyceride makes kind of an E shape. Okay, the letter E, a capital letter E. So you have triglycerides. This includes fatty acids that can be saturated or unsaturated. Triglycerides are also the main ingredients in oils and animal fats. So if you look at bacon fat, fat on chicken, fat on say pork chops or ham, steaks, for example, all of those are going to contain triglycerides. Your vegetable oils, your cooking oils will also contain triglycerides. Okay, phospholipids. This is the second type of lipid. Phospholipids are going to actually be found within the cellular structure of our bodies. Think of all of the trillions of cells that make up our bodies, from the organs, from the blood, from the vessels, from the skin, your hair, all of the cells that make up your body. Each cell is surrounded by that cell membrane. We've kind of talked about cell membranes in the past. We're going to get to those in the future as well. Well, that cell membrane is made up of something called a phospholipid bilayer. That phospholipid bilayer, phospholipid being the second type of lipid, bilayer means two, so it's two-layer. So this is a two-layer phospholipid membrane that surrounds the cells. Here's a good picture, kind of a side shot. If you're looking at the phospholipid bilayer, the cell membrane, notice here is one layer, phospholipids. Here's the other layer, phospholipids. And when we get into cell membranes, we'll kind of get into this in more detail. But the way this is laid out, these phospholipids, it has a hydrophilic head, okay, which means it likes water, it likes to interact with water, it allows water to pass through it, and those types of things. It also has a hydrophobic tails, the center part, which doesn't like water. It kind of repels water. So the way it's built, it allows water to pass, but it also makes sure that not too much water passes. It's a very, very well built membrane, this phospholipid, to help regulate the amount of water. So not too much, but enough can make it into the cell. So this is the second type of lipid, phospholipids. The third type, steroids. Okay, steroids, they're part of cell membranes. They help maintain fluidity of the membrane, which means they help maintain the fluid nature, the passing in and out of the different fluids that help the functionality of the cell. It helps maintain this, especially during cold temperatures. Things move a lot slower in cold temperatures, so it helps in that process. Cholesterol, a type of steroid, cholesterol, can build up in blood vessels, okay, and block blood flow. So steroids are the third type of phospholipid, and one example would be cholesterol. Another example would be the steroid hormones, such as the sex hormones, testosterone and estrogen. Testosterone produced by the testes for males during puberty, or starting at puberty, and then estrogen produced by the ova or the ovaries of the females. That also starts at puberty. So you have two types of steroids. Steroid is the third the third lipid, and then two types of steroids. You got cholesterol, and you have steroid hormones. So here's just a picture of how cholesterol is embedded within the phospholipid bilayer to help with that fluidity, especially during colder temperatures. All right, let's look at how triglycerides are built, how they can be broken down. What's that process? We're looking at macromolecules, where you can take single units, single monomers of a macromolecule and put them together, bond them together, right, by looking at this picture, by combining a hydrogen of one molecule and a hydroxide of another molecule. That makes water. We can take that water out through dehydration. It's going to synthesize or make a new compound. This process is called dehydration synthesis. Okay, so looking at these questions, what's being removed is the hydrogen, just mentioned that. From the glycerol, what's being removed from the fatty acid is the hydrogen. What do they form? Water. What's this process called? Dehydration synthesis. Okay, dehydration synthesis. Taking these monomers, putting them together, forming bonds, removing water as a process. That's how you build these macromolecules. How many water molecules from a triglyceride? Well, you have to have one here, and one here. There actually should be, obviously, 3, not 2. But nevertheless, so looking at dehydration synthesis, building of those macromolecules. All right. What elements are in lipids? Well, let's review carbohydrates. Remember the 3 major elements in carbohydrates: carbon, hydrogen, oxygen. Looking at this triglyceride, what 3 elements do you see? You see carbon, you see hydrogen, you see oxygen. Do you see anything else in this triglyceride? No. So carbon, hydrogen, and oxygen are the 3 elements that help make up lipids. So our carbohydrates and lipids made from the same stuff, just rearranged in different order. All right. So that wraps up our lipids topic. Quick review. Lipids are long-term storage of energy. 2 categories. You have saturated and unsaturated. One's bad, one's good for you. 3 types of lipids. You have the triglyceride, which is the very common type. Okay, it makes an capital E shape when it's formed. Tri meaning 3 fatty acids, and then your glycerol. Then you have the second type, which is the phospholipids, helps in the membrane functionality. And then the third will be steroids. Okay, then your 2 types of steroids would be cholesterol and then steroid hormones.